Welcome to ASM Live, the internet talk show live from the American Society for Microbiology General Meeting in Denver, Colorado. Today we're going to speak with our guest, Leanne Jaykus from North Carolina State University. We'll be talking about norovirus and how norovirus influences our health, how we can detect it, and many other interesting aspects of this virus that has a huge impact on society. I'm your host today. I'm Stanley Malloy. I'm the Dean of the College of Sciences at San Diego State University and the chair of the editorial board of Microbe Magazine. I'd like to ask anyone in the audience who has questions to please come over to the microphone. Anytime you have a question, it's important that you go to the microphone so our listeners in the ether can hear your question. And those of you who are out there on the internet, you can send questions via Twitter to hashtag ASM2013. So Leanne, it's nice to talk to you today. Could you tell us a little bit about norovirus? What is it and why is it so important?
Noroviruses are now considered the leading cause of acute viral gastroenteritis, certainly in the United States and probably around the world. And the current CDC estimates are about 21 million cases in the United States every year, which is a lot when you look at our total population. My particular interest is in foodborne transmission routes, so eating food that's contaminated with norovirus. But the reality is that noroviruses can be transmitted by lots of different routes, so food, water, and probably most important, transmission between people, particularly in homes, institutional settings like elder care facilities, cruise ships. Noroviruses are what cause cruise ship outbreaks.
So what are the symptoms? What happens if you get infected?
Well, the hallmark symptom of a norovirus infection is actually vomiting. So most people present with vomiting. Usually, certainly nausea associated with that. Frequently, abdominal pain and diarrhea as well. It's actually what I believe my mother used to call the 24-hour bug. So you are really sick for 24 or maybe 48 hours. You feel like you're going to die, but you almost never do die. Most people do recover completely from the disease.
Mm-hmm. And how is it transmitted?
Well, we believe that probably 75% of transmission is certainly person-to-person spread. So how does that happen? Well, there's clear fecal-oral spread, so from the feces of an infected individual into the mouth of an uninfected individual, changing diapers or something like that. But over the last 5 to 10 years, there's been a lot of evidence that the virus is actually shed when people vomit, and people believe that some of that vomitus becomes aerosolized. And because the viruses are so tiny, they can actually stay in the environment for long periods of time. And then somebody can inhale that contaminated air. It gets in your, in your mucous membranes, comes down into the GI tract, and you get infected.
So what about foods? Is it the same fecal-oral contamination that leads to transmission in foods?
In foods, it's almost always fecal-oral contamination. The most common foods that are associated with norovirus transmission are called ready-to-eat foods. So those are foods that have a lot of human handling, that do not have a terminal heat treatment prior to consumption. However, we know that molluscan shellfish can become contaminated by fecal matter in the waters, and fresh produce, we're not quite sure, possibly irrigation waters as well.
Mm-hmm. And so decontamination, what about decontamination?
Decontamination. Well, we look at decontamination really in two ways. One is on surfaces, and the second is on hands. So let's talk about hands first, because a food handler that has fecal contamination on their hands pretty much needs to get rid of that. And from a regulatory standpoint, at least in retail institutional establishments, that's actually regulated by the U.S. Food Code, which has very strict hand washing requirements. Now, compliance with those hand washing requirements tend to be abysmal. So even though there are regulations, a lot of people aren't doing that. And thorough hand washing is really probably the best control. There's been a lot of discussion about hand sanitizers, particularly the alcohol-based hand sanitizers. And the bottom line is that we have a lot of evidence that alcohol as an agent has very little efficacy against noroviruses. Now, formulation matters. So what other agents are in the formulation of an alcohol-based hand sanitizer may actually increase its efficacy, but we should never rely on those hand sanitizers as our sole control mechanism for hands. For surfaces and for and for food products, it's a little bit more complicated. CDC recommendations actually are certainly over 1,000 parts per million chlorine, and maybe even between 2,000 and 5,000 parts per million chlorine. For those of you who don't know, that's a huge amount of chlorine, and so that would be what would be recommended for cleaning up a surface that you think might be contaminated.
That's a lot of surface on a cruise ship to cover.
It's a lot of surface, and think about carpets or upholstery. I mean, and nobody's going to use that much chlorine on carpets and upholstery.
But hand washing, I can't believe this is still a problem, because every time in the bathroom it says employees must wash hands, so it should be solved, right?
It should be solved. The evidence is that compliance with hand washing, particularly during very busy periods like in a retail food establishment, is probably, well, it's probably somewhere around 25%.
Now, your work is very largely focused on some of the agricultural-based transmission. Could you tell us something about how you're approaching this problem?
Well, we're approaching the problem sort of in a two-pronged way. So one is to look at ways to prevent or eliminate contamination in the agricultural environment or in a food environment. And the second way is sort of getting the word out to individuals who might be involved in either potentially contaminating food or in helping to control that contamination. So from the first standpoint, what do you do in the agricultural food production processing end of things? Certainly good agricultural practices, which are mandated by the new Food Safety Modernization Act of FDA. We're in the process of including information on noroviruses in those documents that will really go widely to the produce industry in general. Most of the produce industry, particularly in the production end of things, don't really, I mean, they think of E. coli 157 and perhaps salmonella, but they don't think of noroviruses. In food processing, it's a little bit more complicated because almost all of what we use in food processing to control bacterial agents really don't work on noroviruses. So people have looked at things like ionizing radiation and heat. They appear to be pretty heat stable. Certainly not as heat stable as a spore, but pretty heat stable. High pressure processing, which is an emerging technology, kind of works to an extent, but probably shouldn't be relied upon for elimination, at least at the pressures that would be necessary to retain the sensory characteristics of the foods. And then there's the whole issue of better hand sanitizers, better surface sanitizers.
So it sounds like what's really needed is an effective way to detect norovirus so you can prevent it moving into the food stream, since it's going to be very hard to treat some of these foodstuffs in a way that will eliminate the virus.
Right. I think that ultimately prevention is going to be our major goal. And detection is one arm of that prevention. Unfortunately, there's a couple of issues relative to detection. So we do have good PCR-based methods for detection, or I should say reverse transcription PCR-based methods. However, for clinical laboratories, they've been a little reticent to actually embrace those technologies. And so there's still this interest in using ELISA-based assays or some of these other methods. And unfortunately, we can't develop antibodies that are broadly reactive to all strains because there's so many strains. So that's really impacted our ability to develop clinical diagnostics. And there's only one clinical diagnostic that's licensed in the United States, and it's only licensed for use in outbreaks. So if you go to the doctor and want to get diagnosed, there isn't an official test.
You know, one of the things that we haven't said is that this is an RNA virus.
Right.
A very important thing, because you mentioned RT-PCR, and that's why it's so important, just for anyone listening. But that also is related to why there's so many different viruses in this same family with unique properties, because of the high mutation rate and variation within the RNA viruses. Correct?
Yeah, there's actually a lot of study going on in this area right now. And what has happened with noroviruses, to sort of summarize, we have two groups, and within those groups we have what we call genotypes. And there is a strain that tends to sort of be the predominant strain at any one time. That's usually called the outbreak strain. And because of evolution and mutation, we get a new outbreak strain about every 4 to 6 years, and that results in a peak of infection. And that's all about evolution and mutation. And interestingly, this year we have a brand new outbreak strain.
That's interesting. You know, one of the things you often hear is that you do not get long-term immunity to norovirus.
Right.
Is that it changes often enough that you're not resistant to the next outbreak strain? Or in fact, do you not get long-term immunity to the one strain that you're infected with?
That's a really good question, and we actually don't know the answer to that. We do know that the strains do evolve, the outbreak strains do evolve to be able to infect a new population of susceptibles. So it is quite possible that if you were infected with one outbreak strain, you might be resistant to the next outbreak strain. Or actually, no, you might be susceptible to the next outbreak strain. The evidence appears to be that the degree of protection you get from infection really is short-lived. So maybe a year. There's probably huge individual variability, but there have been human challenge studies where they've challenged people a relatively short period of time after they've been infected with the same strain, and they've gotten sick again.
Sounds like a fun study to participate in.
Yeah. Usually they have college students do it and they get paid for it.
So I would like to remind anyone in the audience, if you have a question, to please come to the microphone. And all of you out in the internet world, if you have any questions, please send it to #ASM2013. So I think we have a question coming from the audience right now.
Great.
Hi, Leanne and Stan.
Hi.
Thank you very much for this. Joan Rose from Michigan State University. You know, Michigan's just had, many, many years of, I think 1 year it was 300 outbreaks or something. It was just crazy. And some of the most recent ones are occurring in these water parks that are actually associated with the lake. I'm wondering if you can comment on recreational transmission and also its survival, because they closed down the park. They reopened it several days later, and of course it was sand, and the water's recycling, and they still got cases. And so I'm wondering if you could comment on recreational outbreaks and also survival, what we know about survival. Thank you very much.
I can't comment a whole lot on recreational because that's sort of out of my area of expertise, although Joan is right, there certainly have been a lot of instances where that's occurred. It's probably a couple of things happening there. Certainly fecal contamination in water. I mean, even if you're in pools, which really isn't recreational, but I suppose it is, the amount of chlorine you would have in a pool would not impact the virus. And there's probably also a big aspect of infectious dose. So it doesn't take that much virus to make somebody sick. The current thought is the infectious dose is somewhere between 10 and 20 infectious units. the second part of the question, remind me, was survival. Yeah, we have a lot of evidence from our lab looking at environmental survival just on surfaces. And we're seeing maybe a 2-log drop or something over a month on a surface. And there is quite a bit of data in water that you're looking at months of survival with minimal loss over that period of time.
This is a really important question because in addition to the public health impact, there's an enormous economic impact when you have to close down cruise ships, when you have to close down recreational areas. And so I would hope that this is an area people would be investing in a lot to try to figure out ways to counter this kind of problem. And to think, instead of just the infection being associated with people or plants, to realize the importance of thinking more broadly about nature and the distribution in nature. But that comes back to one of the things I'd like you to talk about. So you're part of a big consortium that brings together different people to attack this problem. Could you comment on the consortium and how different people are impacting the solution?
Yes, I'd be happy to. So I lead a consortium called the USDA NIFA. NIFA stands for National Institute of Food and Agriculture, so it's a research arm of USDA. And we call ourselves the Food Virology Collaborative, or NoroCor for short. Basically, we're a group of scientists that are tackling the foodborne virus problem. And we've divided our efforts into 6 major, what we call cores. One is molecular virology, so looking at molecular aspects, particularly trying to cultivate the virus in vitro because we can't cultivate or we can't grow the virus in cell culture. The second is the detection core, where we're trying to develop detection methods for both clinical and food and environmental samples. The third is the epidemiology and risk assessment core. The fourth is prevention and control. And then we have an extension and outreach core where we're really working with our stakeholders to try to meet their needs to control noroviruses. And then finally, a capacity building core where we're trying to train the next generation of food virologists.
That's great. You know, I'd like to come back to detection for a minute because you just said a short while ago that 10 to 20 viruses is enough to cause an infection. So any detection method that you're going to be using in a lot of these situations has to be exquisitely sensitive, even in a situation where you may have a lot of soil or sand or plant material or whatever associated with it.
Right, and that actually brings up this issue of detection in a clinical sample versus detection in an environmental sample. So if somebody's clinically infected, they're shedding huge amounts of virus. And some literature, depending on the virus strain, actually state up to 1 billion particles per gram of feces. So for clinical detection, the numbers aren't such a problem. For food and environmental samples, you could have a food sample of 25 grams or a water sample of liters that might only have a few viruses in it. And that complicates detection tremendously.
So one of the interesting things in thinking about norovirus is it seems, as somebody who doesn't work in this field, it really seems like this has been increasing over the last couple of decades, where when I was a kid, we didn't hear so much about it. Maybe we just didn't know what it was. But cruise ships, I mean, these are big events that we're hearing about now. Is it evolving? Is this a disease that's becoming more prevalent than it used to be?
Well, I do think there might be an increase in prevalence due to the emergence of these epidemic strains. We certainly are recognizing the emergence of the epidemic strains over the last 20 or so years. However, I think, and my colleagues at CDC think, it's largely because we can now detect using molecular methods, and there's a lot more awareness about this particular organism. So I think it's a combination of both, but mostly it's increased in awareness and better ability to detect in clinical samples, even though we don't have a licensed method, so to speak.
You know, one of the things that I think a lot of people who are concerned about foodborne disease would love this idea of a detection method that could be on a chip that you could spread out somewhere in the environment, and it would, if there was a positive hit, it would send a signal back by radio waves or any other mechanism and allow people to broadly detect the virus in the environment. Do you see a potential future of that?
Well, it's interesting that you asked that question because we had a stakeholder session about 6 months ago, and we really asked our stakeholders, and this includes people from grocery and retail sectors, as well as from sort of nursing homes and cruise ships, what the single most important control measure they could have, aside from hand washing, which clearly isn't working. And they said they really wanted a point of use device where you could do exactly what you're talking about. Now, I think we're a ways away from that, particularly for surfaces and things like that. But I do think that is on the horizon, and we hope that we would have something like that in the next 10 to 15 years.
That's great. We have another question from our audience.
Hey, Doris.
Hi, Leanne. This is Doris D'Souza from the University of Tennessee, Knoxville. I had a question regarding detection since we were talking about it. How do you see next generation sequencing evolving for detection of viruses as they evolve? And the second part of the question is, for control methods, as you mentioned, what about contact time? How does contact time play a role as well? So that the audience would know. Thank you.
Okay, so the first question is about next-gen sequencing. I think that, for instance, in our collaborative, we have individuals who are working on developing microarray technology for either, one group is working for detection purposes and the second group is working for genotyping purposes. I think next-gen sequencing really is going to be most applicable to fecal samples from infected individuals, just because you have such a high viral load in those. And in point of fact, we're also working on biosensor technologies to try to address that issue. In terms of contact time, that is a complicated question because it would depend upon your agent that you were using for an activation. There is evidence that contact time does make a difference in most instances, but for something like ethanol as a disinfecting agent itself, contact time doesn't seem to make a big difference. For chlorine, it does, so it really depends.
So, Leanne, one of the other things that I know you're very interested in is this whole business of transmission by aerosols, right? So could you just give us a story of any cases that really emphasize the importance of transmission by aerosols?
There are really two stories that I think are really fascinating. The first is there is an instance where somebody had a norovirus infection and they vomited in a restaurant setting, like a banquet hall, and there were tables all around, okay? And they did an epi study where they looked at the attack rate after the vomiting incident, and they found that the attack rate was proportional or inversely proportional to how far away the people were from where the vomiting incident occurred. There's also another really fascinating outbreak that occurred in a hotel where somebody vomited on one floor of the hotel with a norovirus infection, and then there was evidence of transmission through the ventilation systems to other floors. And so clearly that has to have been aerosolization.
The very compelling stories. If I recall the story from the banquet hall, people at the same table, it was something like 90% of them became infected.
Yeah, I think it was 80 to 90, yeah.
And when you moved across the room, it was still 25%. So that means that the aerosolization is pretty powerful. And I think it ties again to this idea of the low dose required to cause disease, right?
It does, and so if you only need a few virus particles and they're floating around in the air, you certainly can be susceptible to infection. And it actually is a big instance. So for example, airplanes or the airline industry is now beginning to be concerned about this. And the other is that many retail food establishments or nursing homes are really concerned about cleaning up vomiting incidents and how big is the radius of impact in the case of a vomiting incident. And we don't have a lot of data on that.
It really gives you a different perspective when you think about that ability to be transmitted by an aerosol like that.
Well, just from a personal standpoint, if I walk into a restroom and I see that somebody's vomited, I certainly walk out. And if I find out later on, you know, like I use the restroom and I look down and I see it, then I hold my breath as long as I can.
Yeah, and if the person at the dinner table with you vomits, it's probably a good idea
I'm
to
out
go
the door. No, it's not.
So the comment was, it's not so easy on an airplane, but at least there's a little bit of time before you actually develop the symptoms. So you probably aren't going to get sick while you're on the plane if you're the secondary person.
Although I believe there was, like a few weeks ago, it was associated with a tour, and the pilot actually refused to take a group of tourists back to Europe because they had vomiting and diarrhea.
It wouldn't have been a very pleasant flight.
No.
Well, with that, I would like to thank you very much, Leanne. It was a really interesting discussion, and I think this is an area that has really broad impact and a lot of potential for the future in studies. I think expanding out to have better ways of detection and understanding the transmission better, simply informing people, and understanding this big question about survival of the virus in nature and what we can do to thwart that. It's going to be very exciting for many years to come.
Yeah, and I'll just put in like my plug as an academician. You know, I think this is a really great opportunity for young scientists. There's a lot of both basic science and applied science, and I think ultimately they can make a big impact on a very important disease that has both public health implications as well as significant, as you said, economic implications.
Thank you very much for joining us for this episode of ASM Live. We welcome you back to another episode later in the show.
Explore the fundamental role of microbes in the natural history of our planet with Microbes and Evolution, the world that Darwin never saw. Available at estore.asm.org.